Good morning, Professor. Am I audible?
No.
Hello. Am I audible to you, ma'am?
Yes. I'm here ready now. Thank you very much for inviting me.
It's completely our pleasure, ma'am. Okay. Good morning, delegate. Good morning, participants. I'm Kiran Suryamshi, lecturer from SMBT Institute of Pharmacy and coordinator of ICTS 2021. Before we start with the today's session, let me take a pleasure to introduce our eminent speaker, Associate Professor Dr. Farahida Mohammed. Mauryan received BSc in Pharmacy from Strathclyde University in 1999. Used to work at the community pharmacy and government hospitals before decided to pursue PhD in 2004. She obtained her PhD in 2008 from Strathclyde Institute of Pharmacy and Biomedical Sciences, Scotland. She has been leading various research projects funded by various agencies and ministries. She had also received industrial grant to lead and coordinate GMP associated trainings. She had participated in more than 30 international conferences, has more than 30 publications, and had supervised more than 60 postgraduate masters and PhD students. While contributing in teaching and research, she is also responsible in managing IKOP. Two of her research products have been commercialized, and she had become one of the finalists for the award of Malaysian Commercialization Year organized by MOSTI in 2017. In 2019, she received the first commercialization award during IIUM Takrim Day. In 2020, she has been invited as a speaker for scientific conference organized by Wahid Hasyim University, Indonesia. Currently, she has been appointed as external examiners for undergraduate programs at International Medical University, Malaysia and UNISA. Three more of her research products will be launched by IKOP this year, with projected to increase IKOP's revenue by 30 to 50%. So that was the shortest introduction of our today's speaker, Dr. Farahida Mohammed. Without wasting much more time, I would like to request her kindly start with his session. Thank you so much. Ma'am, you can start with your session.
Yes. Okay. Can you show me my slides, please?
Slide share. Yes, sir.
Yes, sure.
All right. You will, okay, start from the beginning. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah rabbil alamin wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma salli ala sayyidina Muhammad. Okay. Thank you very much to the organizer who invited me to present in your platform. I'm very honored to receive the invitation, and today I'm going to share with you one of our research project. It was conducted with the help of an Indonesian student, where she has attached to our Kuliah of Pharmacy, Faculty of Pharmacy Malaysia, for 3 months. And this project just been completed within 3 months' time, which is very excellent student. who has helped us. Okay, so this topic today is going to share on the hepatoprotective effect of paracetamol honey suspension. Next slide, please. Next slide. All right. Okay. Hang on. Sorry. The outline of the previous slide. Sorry. Okay. The outline of topic today is I'm going to show you some introduction and the concept on halal and Thayyiban fusion medicine, which is today the conventional the fusion medicine. We use the paracetamol, which is the conventional drugs, together with the traditional drugs, which is traditional medicine honey. To the in vivo testing on hepatoprotective effect in rats and some results and discussion. Okay, next slide. Next slide, sir. Next. All right. I think everyone know what is acetaminophen. Previous slide, please. It is also called paracetamol. Okay. Sir, please previous slide. Can we go to previous slide, sir? There are various types of dosage of paracetamol. It is marketed in few type of dosage forms, syrup, suspension, and as well as tablets or caplets. Okay, and not only the single drugs, it appears as single drugs, but also marketed as fixed dose combination drugs with other active ingredients apart from paracetamol. So, for example, you can have when you have an headache, say for example an elderly patient who is on chronic medication for muscle relaxant, and they don't really understood that their own existing medication consists of paracetamol, okay, with muscle relaxant. So when they have headache, they tend to take another paracetamol tablet. So this can easily cause drug overdose of paracetamol. So actually, next slide. Next slide, sir. Okay, here. So basically, if you can see here, I'm sharing with you a few types of drug combination and paracetamol alone. For the paracetamol, we can have syrup, formulated as syrup, suspension, and also tablet, 120 to 150 milligram, etc., etc. And even the tablet paracetamol itself has the dosage strength for children as well as for adults. And then the combination of paracetamol with other drugs like caffeine, it is used for menstrual pain, and like paracetamol plus orphanadrine citrate, it is used as muscle spasm. So basically, easily this PCM is a highly classical drugs but widely used worldwide. Not only appear as a single API, but also with combination. And the market size for this PCM is USD 1 billion annually. All right. So next slide, sir. So basically what we want to tell you here is that paracetamol is very widely used, and overdose of paracetamol easily happen. Okay. And the problem with the syrup or liquid formulation in suspension, they usually contain about can contain sucrose or sugar up to 80%. So that will be detrimental to our children here, like shown in the picture, actually my children. Right. They have the reason to add in this sucrose or the sugar in paracetamol syrup formulation is to mask the bitter taste of paracetamol. So, and not only paracetamol, but other sugar or other syrup or liquid formulation, they usually have this high amount of sugar. Okay, this high sugar content in pediatric liquid medications, what we concern is its potential detrimental effect, carcinogenic on that cause dental caries, and this is detrimental to the healthy growing teeth of our children. So what happens if they have frequently have the coughs and colds? For children, their immunity still developing. So this is our concern. Next slide, please. So we inspired by looking at this kind of problem, we formulated an alternative form of paracetamol suspension by substituting the sugar with honey. So it becomes something like synergistic. Okay. So another problem with this paracetamol, when we referring to the other type of dosage form like tablets, okay, the tablet existing marketed paracetamol tablets, it doesn't have any added value, just have give a quick effect on the headache, for example, or as analgesic. Okay, especially for chronic disease, and recently this UK, the NICE UK, which is the National Institute for Health and Care Excellence, NICE, the NIH, National Institute of Health of the UK organization, that usually come up with guidance, okay, clinical guidance for something. So they come up with these painkillers such as paracetamol and ibuprofen. They said should not be prescribed for chronic pain because they do more harm than good. So if we can have some alternative, or not only paracetamol but paracetamol with honey, which is the traditionally used medicine, we already know honey is very safe. Honey is has antioxidant effect, has flavonoids content, phenolics, which can give a synergistic or a wholesome benefits of the medicine when we use this paracetamol with honey. So it can also reduce the risk of overdose. In fact, even the acute overdose of PCM can be mitigated if we have an antioxidant administered concomitantly together with this paracetamol form. So risk and the USA. I'm not sure about India scenario, but USA usually they have their established reporting, adverse effect reporting. That's why I could find some information regarding this risk of or the prevalence of overdose of PCM. Next slide, please. Okay, as I mentioned just now, the problems with the existing formulation of paracetamol is the sugar content. Not only my concern or our research team concern, but also other people as far as UK researchers, they also, or the nutritionists of these UK people, they also identify the bad effect of sugar content that can cause their dental concern with commonly used medicines. Okay, so in this example, they are dentists. Can I have my slide? They are dentists, and they find out that commonly prescribed medicine for dentists to their patients contain high amount of sugar, and because they're treating patients or children with dental problems, they found out that it is very difficult to cure these problems because and it is related to the high sugar content of the pharmaceutical syrups that they prescribe, which is paracetamol. And not only that, the antibiotics suspension also contain high sugar. Sir, can you give me my slide? My slide doesn't appear on my screen. So the next The next slide. Hello, sir. Can you show me my slide, please?
Yes, Professor. Can you tell me which slide number do you want?
Okay. After the mail slide number.
Can you please tell us which slide number do you want?
Maybe number 6.
6. Okay.
Number 6 done, and number 7 on sugar content, right? Okay, so next slide number 8. Okay, this table is just to show and share various type of possible possible pharmaceutical syrups, including all the antibiotics type, which the API themselves are very bitter. Similarly to the acetaminophen or the paracetamol, they are the bitter type of API. That's why for formulations in syrup dosage form, we need to add a lot of sugars to mask the bitter taste. So the various sugary pharmaceutical syrups and suspension, and what we found out, the article mentioned that average sugar content is 4 gram per dose. 4 gram is a lot. 1 spoonful equivalent in 5 meal. When we give our children, they has to take 4 gram of sugar. Okay, next slide, sir. On introduction and pain. So not only the sugar content which is high, but the paracetamol itself can easily can cause drug-induced liver injury, especially when taken by elderly patients. Okay, our elderly patients, they didn't understand, they didn't aware on existing medication. Sometimes they are taking paracetamol with drug combination, and then when they have headache or fever, they tend to just take another tablets of paracetamol alone to cure their fever. Okay, next slide, next slide number 10, Journal of Hepatology. Okay, this Journal of Hepatology, they also mentioned about drug-induced liver injury, and they found out that acetaminophen can cause hepatotoxicity, which is the most common cause of acute liver failure, ALF, in the US. Even a single overdose can, of the unintentional case, happen, and happening even for the doses that they are taking, the daily doses, recommended daily doses, 4 to 10 gram per day. This also has been reported to cause acute liver failure in the US, right? Other issue with this paracetamol is as I mentioned, the concomitant drugs. Okay, factors such as concomitant drugs, systemic illnesses, and some patients, they are also chronic alcoholic abuse patients. So when they take paracetamol with when they are already an alcoholic, their liver already sick, their liver already damaged. Okay, when they take paracetamol, which has the hepatotoxic effect, this can aggravate their symptoms or their hepatotoxic effect. Right. Can we go or move to slide number 11? Okay, slide number 11 is showing example of acetaminophen bottles. Okay, an example of drug associated with intrinsic and versus idiosyncratic DILI, which is the drug-induced liver injury. Okay, there are many type of drugs that can induce liver injury, and particularly we are concerned with the acetaminophen. And for the concept purpose, proof of concept, we are taking acetaminophen as an example in our research because acetaminophen is easily to get. We don't need any special license to purchase, or we are easy to control in our inventory. So that's why we are taking acetaminophen as one of example to show that when we combine with traditional drugs such as honey, we can reduce or minimize the effect, the bad effect of this drug on our organ. So today on our liver. Okay, can we move to next slide, sir? Slide number 12. Okay. All right, slide. This slide is showing WHO Sustainable Development Goals. Okay, I'm sure everywhere in this world we are aware about these WHO Sustainable Development Goals. There are 17 of them, and what related the one related to our research area, our academician profile is number 3 on the good health and well-being, number 9 some innovation, and number 16 partnerships for the goal. Okay, partnerships for the goal actually meant not only between the academician with the industry players, but also we are together with all the publics. If the publics can listen and we can increase the awareness on this fusion medicine, the conventional together with the traditional medicine, which can have a synergistic effect, this would be great. Okay. And this because IKOP is we pronounce as ICOP. Okay, it is we are our vision is to produce halal and thayyiban medicine, but because especially on research base, we are targeting this fusion medicine, the halal parts and the thayyiban parts, but focus on the fusion medicine, not on the single API conventional, but we are targeting to produce a better medicine, which is fusion medicine, which to us, it should have this kind of attributes, WISE, wise. It should have a wholesomeness attributes, medicine that care and cure and actually treating the disease. It should have an I aspect, innovation aspect of medicine. It should also have the S, sustainable and synergistic effect. Sustainable, which means the medicine is easy to get. The medicine also is actually eco-friendly. Okay, and it produces synergistic effect when combined together. And E, it should be equitable, basically referring to the price. Later on, the price will not be so expensive. It should be accessible by anyone, whether they are farmers or they are lawyers. These are kind of wise medicine we are looking forward to produce in the future. So these are the characteristics of the fusion medicine. Halal and thayyiban, fusion medicine we are in our mind, in our vision to produce. Okay, sir, next slide, please, sir. slide number 13. Okay, this slide showing our artwork of the products. Paracetamol, honey suspension, and we are using paracetamol, the real raw material paracetamol powders. Not usually sometimes when we do research, we tend to buy tablets of paracetamol, we crush and we mix together with honey. But we are not doing that. We are buying from the original raw material supplier of paracetamol in the powder form, and then we mix together with honey and some excipients to make it stable. Okay, so this is our formulation. Okay, next slide, sir. Okay, this the paracetamol is very conventional drugs. The approved indication is as antipyretic and analgesic. For our formulation, we actually already done an animal study to compare the synergistic effect with paracetamol alone. Okay, when we compare, the synergistic effect is being demonstrated with our fusion medicine. We got drugs, a medicine that can reduce the body temperature better and prolong reduction in body temperature, as well as the analgesic effect, prolonged anti-pain effect when we tested with animals. Okay, but today I'm going to share with you the hepatoprotective effect of this paracetamol honey suspension. Okay, next slide, sir. Okay, honey. Honey itself is traditionally, I think all of us know what is honey. Can be various type of honey can be produced depending on different type of flowers. The bees take the nectar of that flowers. So major components, honey actually also available in pharmacopoeia. The USP, BP, they mention about the specification of honey. Okay, so for honey, it has major components as sucrose, fructose and glucose, and it has low glycemic index, traditionally used to reduce fever and demonstrate hepatoprotective activity, antibacterial effects, and anti-inflammatory effects. So we have chosen honey to be the traditional or prophetic medicine to be fused and formulated together with the conventional drugs, which is paracetamol. Okay, next slide, sir. All right. So for today, I'm going to share the hepatoprotective testing that we have conducted in rats. Okay, we use spread dolly albino rats, 24 healthy males, and they are aged 5 to 6 weeks. And we conducted this after we got the approval, ethical approval from our IIUM, International Islamic University Malaysia Ethics Committee. So next slide, sir. All right, after we got these rats, we acclimatized the rats for 7 days with normal feed and tap water. Okay, and then we divide them into 4 groups. Group number 1, each of the group consists of 6 rats, and group number 1 doesn't receive anything. They just receive all normal tap water and normal feed and tap water. Okay, group number 2, they will be receiving they receive the paracetamol with sugar suspension. Group number T2, we call it T2, is going to be treated with paracetamol honey, and the final one is with honey alone. So altogether we have 4 groups. Number 1 is control. We is normal, doesn't receive any intervention or any paracetamol, not honey at all. Okay. Number 2, receive paracetamol suspension sugar one, which is the commercially available paracetamol sugar. Okay. Then we compare with the paracetamol honey, the one that we make, and honey alone. Okay. After 7 days, during the day 8, we give them 2 gram of before treatment. We take some blood samples, okay, just to make sure all of them have a healthy biomarkers that we are targeting, which is the AST and ALT. Okay, AST and ALT is the liver enzymes, which are very important biomarker uh, for for identifying whether our liver is healthy or not. All right. So when we before the day of treating, we take their blood samples, and then after the next day, we give them the paracetamol with sugar, paracetamol with honey, and honey alone. Right. After 24 hours, the dose that we gave them is actually a toxic dose. A dose that from established literature, this dose 2 gram per kilogram body weight can give hepatotoxic effect on the rats with single dose administration. Okay, after 24 hours, we euthanize these animals and we collect the blood. Okay, we have to sacrifice the rats to collect the liver. And the liver, we do the histopathology examination as well histopathology examination. Yes, and that the blood we send out to check the level of ALT and AST, comparing the blood profile before paracetamol intoxication and post 24 hours intoxication. Okay, next slide, sir. Slide number 18. All right, this is just to show you the example of acclimatization. We have the rats put in the cage with normal feeding and tap water. Okay, on the right hand side is where we show or demonstrate the force feeding technique of to the animals. Okay, next slide, sir. Slide number 19. All right, these rats already euthanized, already anesthetized. When we want to collect the blood before the treatment day by cardiac puncture. Okay, can we move to the slide number 20? After the treatment, we have to, after 24 hours, we have to sacrifice the rats and take out or remove the liver for further histopathological examination. Okay, can we go to slide number 21 on the results and discussion? All right, here the graph on your left is showing the rats food intake from day 1 until day 10. Day 10 where we sacrifice them. Okay, as you can see, during day 1 until day 7, during the acclimatization day, they are very healthy. They take food regularly, and when we start to do the cardiac puncture and take their blood before a day before treatment day, they start to they suffer from stress, a bit of stress, and that's why the food intake reducing on day 8 until the day 9 where we where we give them by force feeding the paracetamol suspension, and day 10 we sacrifice. Okay, so on the right is on body weight. Body weight doesn't really show any effect even after cardiac puncture. Okay, but from day 1, of course, after they taking several days after taking food, they start to gain weight. Okay, can we go to next slide, please? Slide number 22. Okay, after intoxication on day 8, we sacrifice them 24 hours after the single dose of toxic dose paracetamol. Okay, then we take the blood and we measure the ALT and AST. Okay, ALT and AST. Oh, sorry. Okay, this figure actually showing you the ALT pre and post. Okay, the similar diagram we obtain for AST as well. So for the ALT liver enzyme, as you can see here, pre-treatment, the blue graph, the blue bars, all of them are not statistically significant. All same alphabet. Okay, so basically during before treatment, they are all very fairly healthy and have same ALT level. Okay, after the treatment day on day 8, when we take their blood and measure the ALT, we can see here this this ALT level for paracetamol sugar suspension becomes tremendously increased. Okay, PSS is the paracetamol with sugar formulation, which is the normal formulation. At 2 gram per kilogram body weight, their liver damage. Okay, the hepatotoxic happen with this normal sugar paracetamol formulation. In contrast to in contrast, the formulation that we formulate with honey at same strength, 250 milligram per 5 ml of paracetamol suspension, but we add in honey, substitute all the sugars with honey. Okay, about 70%, 60 to 70% of honey is present in the formulation. So it shows that this presence of honey can greatly reduce all the toxic effect of the liver. Okay, PHS here um, is the honey, paracetamol honey. Okay, when we compare with honey alone, yeah, alhamdulillah, honey alone of course doesn't cause any hepatotoxic effect on the liver. It's very safe, right? Okay, this is ALT on the left and on the right is AST. Similar trend, ALT and AST. They are the groups, the animal groups with that is intoxicated with paracetamol sugar suspension. Their liver profile become very bad. Their AST and ALT increase. Okay. All right. Can we go to slide number 23 on liver histopathology? Okay, this liver of the animals of the rats shows that we have 4 liver sectioning here. The first one A is the liver from the control group, which doesn't receive any honey, doesn't receive any paracetamol. Okay, they show a very healthy liver cells, right? On the B, picture number B shows the damaged liver from the groups that receive paracetamol sugar suspension, the normal one in the market. At the 2 gram dose, single dose, their liver become distorted and the architecture is not normal. The CV, which is the central vein, distorted as well. And this indicate their liver injury of the rats that receive paracetamol with sugar suspension. Okay, the liver of the rats that receive paracetamol with honey, our formulation, they show a little bit of toxicity effect, but their CV, which is the central vein, is protected. Okay, there are also so almost near to normal healthy liver. Okay, the liver of honey groups also normal and very similar to the group of rats that doesn't receive anything. Okay, so basically here from the liver histopathology data, we can safely mention that the presence of honey in paracetamol suspension can protect or reduce the toxic effect of paracetamol. At 2 gram dose, which is the toxic dose of paracetamol, presence of honey able to minimize the toxic effect. Alright. Can we go to the next slide, please? Slide number 24. Okay, this slide number 24 is to show you the paracetamol mechanism of toxicity. Okay, basically paracetamol, when we take paracetamol, it will be converted to NAPQI, and when at toxic dose, our own glutathione will be depleted. They cannot convert and cannot render it safe, but it will so complex pathway here, it will finally cause cellular death. Okay, so paracetamol poisoning is happening sometimes in hospital. We have the not sometimes, in the hospital itself we have paracetamol blood monitoring. Whenever there is adverse effect of patients being admitted, even pharmacists has to quickly come to the hospital, even at night, to settle this issue. Okay. All right. So can we go to the next slide, 25? I have 3 more slides. All right. Slides 25, results and discussion. Right. So basically the paracetamol intoxication, as you can see, at 2 gram dose can cause remarkable liver damage. Okay. Indicated by the reduction in the, perhaps due to the reduced oxygen perfusion. So this causes the development of liver diseases, and which progress to the cell death. And as you can see from the histopathological data, it is characterized by congested central vein, dilated sinusoidal fissures, and fatty changes, and loss of architecture of the liver. Okay, as mentioned, it is due to the NAPQI free radicals resulting from the excess metabolism of paracetamol, which interferes with protein synthesis and production in the ribosome. As the results, this kind of proteins and lipoproteins, they cannot enter out of the plasma. They form the triglycerides and stay there and cause the fatty accumulate in the liver cells. Okay, slide number 26, second last slide. Right, honey treated architecture and less extent of hepatocellular hypertrophy, which confirm the hepatoprotective effect of honey. And based on the liver histology of paracetamol honey suspension, the presence of honey in our formulation show protection of honey is there. And when we compare with our results of phytochemicals results, that quantify the phenolic and flavonoid content of our honey, it shows a good corresponding data. It corresponds with the increase in phenolics and flavonoids paracetamol as compared to paracetamol normal sugar formulation. Paracetamol sugar formulation, they doesn't have this phenolic and flavonoids. But paracetamol honey, yes, we do have this phenolic and flavonoids level, and which this can eliminate the radicals, free radicals, it can inhibit oxidative damage and consequent degeneration of the liver tissues. And then in the honey itself, some of the honey has non-protein sulfur-hydryl groups, and because some of the honeys they may contain amino acids and other micro elements. Okay, and this may have a role in the prevention of toxin-induced morphological changes, including the central vein distortion or degeneration in the liver. Okay, so the results, what we obtain here, histopathological results and biochemical level, determining the AST and ALT level. It shows that our the result is corresponding to each other. High level of phenolics and flavonoids give a protection of the toxicity of paracetamol, protection at the liver. Okay, last slide, sir. Conclusion. Right. I think everyone know now that paracetamol can easily be, our anyone can easily get overdose of paracetamol, but the susceptible group is the children and the elderly patients. Okay, risk of overdoses of paracetamol, however, can be greatly reduced if we can concomitantly administer something antioxidant which is very safe, like honey. Okay, but the taking of paracetamol separately with honey, it will cause some lower compliance and will increase the cost of to buy the honey, cost to buy the medicines. So what we are targeting here, we want to reduce the cost, we want to increase patient compliance. That's why we do the fusion medicines. Okay. So the results of this study demonstrated the hepatoprotective action of honey in paracetamol honey suspension that we formulated. And even at the toxic dose that with presence of honey can reduce the animals. So what if we just use the normal dose of paracetamol? So inshallah our liver will be protected well, especially the liver of the elderly patients, which is already worked very hard over the years, especially those patient elderly that has to take polypharmacy. They have so many drugs to take. Their liver already work very hard to metabolize all these drugs. So this kind of protection is should be in place to help them and to protect or to prevent further damage. Okay, the protective effect of this honey may be due to their antioxidant property. And therefore, this paracetamol honey suspension can serve as the newly introduced analgesic and antipyretic drug that have a hepatoprotective effect. Okay, the analgesic or the pain, even the chronic pain, these are very common pain. Most people, I think, maybe 90% of us has experienced pain. Okay, and has experienced headache and fever. So we hope that with this kind of concept, fusion medicine, utilizing conventional drugs or utilizing contemporary drugs, API, we fuse with the established traditional drugs like honey, can give a synergistic effect and can reduce the adverse effect of the existing drugs. And this similar fusion medicine, I think we should encourage and we should advocate the use so that many more studies will be conducted by anyone so it can prosper. And the partnership with the SDG goal number 17 that I mentioned earlier, to achieve the goal with partnership, we need to partner with academicians, need to partner with the industry, and both of us need to do proper education to the public, so everyone are aware with this kind of concept, fusion medicine, and inshallah there are many more people to support this kind of effort. And of course, we must ensure the fusion medicine should also be supported to undergo appropriate clinical trial so that we can establish a more concrete evidence. Okay. With that, I think that will end my presentation. Thank you very much.
Thank you so much, Professor. Before I conclude this session, I would like to ask participants, if you have any doubt, please raise your doubt. We have a chat box, you can type your query over there.
Maybe presentation is very clear, that's why there's no questions. But it's okay, if you have any questions, can always email me. At farahida@ium.edu.my. We can have other this kind of more discussion on this topic, perhaps in the future.
Yes, one of the participants is asking one question. The question is, what about the stability of product? Paracetamol, honey, I think she has written PCME, honey.
Paracetamol with honey. Okay. What about stability of product?
Paracetamol with honey.
Manjula Ruparel, India.
Yes. The question asked by Manjula Ruparel, India. Ma'am, please make yourself unmute.
Okay, sorry. Okay, can you hear me now?
Yes, yes.
Yeah, all right. Okay. So we have conducted the stability at the scale up stage, where we have the plant in our campus. We have a mixing tank, big mixing tank, and GMP certified plant factory. And we have using for this paracetamol honey suspension, and we have condition as well as real-time real-time chamber and accelerated stability chamber at 37 degree and 40 degree, and RH is at 75 RH. So everything is based on the characterization, the particle size, and the 2.5 years. I think the study the study of the stability already reached 3 years, and the product is very stable. No issue with the stability.
Professor, we have one more question from the participants.
All right.
I request Muta sir kindly.
Hello, ma'am. Ma'am, my question is simple. That is, what is the rationale behind using this particular combination? That is combination of herbal formulation with synthetic medicine, that paracetamol and honey.
Okay, the halal parts is to target not only non-Muslim. For all, halal is actually limitless. when we add in the added value of halal, it will give a high assurance to all the Muslim populations that this product doesn't contravene with any religious belief or religious obligatory requirement to take the medicines. So not only halal, but the toiban parts. The toiban is the wholesomeness that we are trying to create. A medicine, a fusion medicine that has the synergistic effect, reduce adverse effect, but have a very good overall wholesome effect, benefits. Like when we have honey, not only it protect our liver, but the immunity of oneself is increased, right? It has anti-inflammatory effect as well. So for like our children, when they have fever, their throat actually inflamed, and we want to reduce this kind of inflammation so that their fever will subside quickly. And the onset of action, we found out that when we fuse paracetamol with honey, the onset is much faster, and the effect is prolonged. So if we have ability to perform a more thorough study, we can establish that this paracetamol honey suspension can can change the prescription behavior of our doctors, can change the posology of the existing paracetamol. Like for example, the existing paracetamol we need to give 4 times a day or 3 times a day, right? Because of the short effect, short duration of the paracetamol to reduce the fever or to reduce the pain. But at animal level, we already tested with paracetamol honey. The presence of honey seems to have a synergistic effect in terms of when they lower the body temperature, it lower and give a prolonged effect of that low temperature, anti-feverish effect, as well as the anti-pain effect. It gives a prolonged anti-pain effect, which is the analgesic part.
Okay, another simple question. Ma'am, as it is a natural material, that honey, whether you have studied about the different sources we have used, maybe the source is maybe from some honey is obtained from some region, and
Yeah.
next we have taken the honey from some another region, so it will affect the composition of that honey, whether it will
Yes.
affect the final formulation.
All right. Yes, I am aware of various type of honey from various region of the world. Okay. um, they will have different quality of honey in terms of micro elements. But in terms of viscosity, we'll also have a problems. But the viscosity parts, we can the manufacturer of the honey need to control. We need to have a good networking with them, discussion with them, that we perhaps, for example, we want them to collect honey, to harvest the honey only between January to March, for example. We need to study this first. Okay, then we need to identify and establish the reproducibility of honey quality. However, the one that we take is actually from a country that have a huge honey productions, and we have already discussed with them, and they have a strict protocol on their collection of honey. So on the viscosity part, we assume we must anticipate there is a wide range of different viscosity specification for honey, and but as long as they meet the specification in the pharmacopoeia, the sucrose must be some range, and the other one is the what they call it. There is one value that a marker that specify in the USP. As long as we follow and our honey fulfill the criteria in mentioned in the pharmacopoeia, then there shouldn't be any issue. But of course, I think not of course, with any other natural products, we need to mention when we do the product registration, we need to mention the appearance parts. Okay, the certificate of analysis of our products, we need to mention on the appearance part that the color would be changed due to the natural products quality.
Okay, thank you very much, ma'am, for your clarification. Thank you.
Ma'am, we have one more question from the same participant, Manjula Ruparel. Does the shelf life of combination of paracetamol with honey enhances the shelf life, or any major observation is seen during experimental studies?
Okay, so far, actually when we do product formulation, we need to start with compatibility study, right? We need to ensure the API that we put and the chosen natural products or antioxidant, whatever traditional product that we will plan to fuse together, already we satisfy with the compatibility at the in vitro parts, which is based on IR.
मलेशिया ते रेंजचा प्रॉब्लेम आहे. आपल्या इंडियात असं नाही. त्याने ते राजेंद्र घेवले काढू नको मला स्पीकर पुढचे काढू नका त्यांचा मेसेज आला.
Hello, Professor. Am I audible?
म्यूट झाले. अच्छा लेफ्ट झाले.
Okay, due to some technical problem, speaker has left this conference.
You will wait for another 2 minutes. We will start the next session.
Let us wait for 2 minutes.
Yeah. All right. Okay. Sorry, sir. Sorry, all of you. Sorry. We are past.
That's completely fine, ma'am. You can continue. You are audible.
In the
रेंज मध्ये नाही आहे सर. messages are there. साडेदहाला आहे ना तुमचं?
We have got the message from speaker that she is going to connect with us shortly. Just wait for her. As soon as she joined this conference, we'll conclude this session. I hope Ms. Manjula Ruparel ma'am is in the list of participants and she is hearing us. Hello ma'am, are you there? We have got the message, we have got the answer from the speaker. Are you there ma'am? The answer is to avoid product instability issue, we must do compatibility testing at the very beginning. It is a pre-formulation stage. Use a DSC or FTIR to check if there is any compatibility issue. I hope the answer is satisfactory and you don't have any doubt regarding this question. Moving forward, I would like to thank you for our speaker, Farahida Mohammed, for enlightening us with his knowledge. The research work that she has done and she is performing assumes to be a sprouting seeds in the mind of researcher which will motivate participant to perform a research in future. Thank you once again ma'am for showering the knowledge on us. Thank you so much. And on the next, I would like to inform you that the next session will start sharp at 10:30, 10:30 a.m. as per our schedule.
IST Mumbai.
IST Mumbai, sorry. before we start with the next session, we have earlier speaker Farahida ma'am. Due to connectivity problem, she couldn't connect with us. Now she is there in our conference. Thank you, ma'am, for enlightening us with your knowledge. The work that you are performing, a research work that you are performing, it would be assumed to be a sprouting seed in the mind of research, and it will be motivate participants in the future. Thank you for showering your knowledge on us. Thank you so much on behalf of SMBT Seva Bhavi Trust, Educational Trust, and second ICTM 2021. Thank you so much, ma'am.
Okay, next speaker will talk.
Now I request Professor Kundan Tiwari kindly introduce our next speaker. Second session of day 2. Please, sir.
तू पाठवले का? तुझा नंबर आहे?
Thank you so much, sir. Oh, very good morning. Good morning, Professor.
Good morning. Hello, namaste.
So it's my pleasure to introduce our today's eminent speaker, Dr. Rajendra Gavli, sir. Sir, now working as an associate professor at Kathmandu University, Department of Pharmacy, Nepal. About educational, so sir has completed postdoctoral research on cancer biology from Jeju National University, Jeju, Republic of Korea in 2011 under the fellowship of Government of Korea. Sir has completed PhD from Koshan University Graduate School of Natural Science, South Korea in 2007 under the fellowship of Government of Korea. Sir completed masterate in botany from Tribhuvan University, Kathmandu in 1998. Also, sir completed bachelor's in biology from Tribhuvan University, Kathmandu in 1996. Now about professional experience. So, sir started professionals from 2001 to 2004 in different colleges affiliated to Tribhuvan University and HSEB. In 2001 to 2002, so work as a faculty in Tribhuvan University Amrit Science Campus. In 2007 to 2008, sir work as a faculty in Asian College of Advanced Studies in Kathmandu. In 2007 to 8, also work as a faculty in Tribhuvan University Central Department of Botany, Kirtipur. In 2008 to date, subject committee member of Department of Pharmacy, Kathmandu University. In 2008 to 2011, work as a lecturer in Kathmandu University, Department of Pharmacy. In 2011 to 2012, sir completed postdoctoral research, Jeju National University, Jeju of Korea. In 2012 to date, subject committee member of biology, Kathmandu University. 2014 April to May, officiating head of the Department of Pharmacy, Kathmandu University. In 2014, work as a coordinator of the organizing committee of capacity building of quality standardization of manufacture of herbal medicine in Nepal, organized by Department of Pharmacy, Kathmandu University. In 2015, coordinator of organizing committees of national workshop on WHO guideline analytical techniques for assessing safety of herbal medicine, organized by Department of Pharmacy, Kathmandu University. Then in 2014 to date, subject committee member of Tribhuvan University Department of Botany. 2011 to 2018, assistant professor Kathmandu University Department of Pharmacy. In 2016 till date, work as associate director Kathmandu University, Nepal. Last but not least, in 2017 till date, sir working as a R&D consultant of Phytomax Care Private Limited, a herbal company of Nepal. Sir also a founder member of same company and currently leading the R&D department of both Phytomax Herbaceutical Private Limited as well as Phytomax Cosmeceutical Private Limited, Chitwan. Uh, Regarding the research interest, so sir has an area of interest in pharmacognosy and phytochemistry, ethnopharmacology, Ayurvedic medicine, bioactives of natural products and their formulation development. Sir also published book Handbook of Pharmacognosy, Navodit Pustak Bhandar, Bag Bazaar, Nepal, and that recommended in the Tribhuvan University, Nepal for BSc Botany. Regarding a research project, Susara has successfully completed more than 8 research projects. Now, more than two research projects ongoing. Then, sir also got more than 9 fellowships. Also got award, that Science Award, Nepal Academy of Science and Technology, Government of Nepal in 2015. Then regarding skills, knowledge, Susar has excellence in pharmacognosy, phytochemical analysis, bioactives of medicinal plant, herbal formulation development, ethnopharmacology, flavor and fragrances. Sir also working as an editorial member for more than 30 journals. Some of like ethnobotany research and applications, pharmaceutical and medical research communication, African Journal of Microbiology Research, etc. Then, sir also work as an external as well as internal examiner of thesis, not only for BPharm, they are working for their MSc thesis as well as PhD thesis. Then regarding research publications, so more than 60 research article as well as review article published. Then sir also worked for peer reviewed proceeding of conference papers. So this is because of the very short introduction of our today's speaker, Dr. Rajendra Gavli sir. So I request you, sir, please continue your session.
Thank you. Thank you, Dr. Tiwari, for a very nice introduction about me, introducing me in this conference. Respected organizing team, and SMBT, Sevavi Trust, SMBT, Diploma College of Pharmacy, Professor Dr. Yusir, and respected participants, all ladies and gentlemen. Very good morning from Kathmandu, Nepal. And today I would like to briefly discuss about the plant-based traditional medicine of Nepalese Himalaya and their formulation development and research work going on in our laboratory. So with this, I would like to start my presentation. As we know that Just a minute. I would like to hide this figure. Mm, hide panel. Okay. Mm, yeah. Uh, if we see the global scenario of the utilization of the medicinal plants, there is the publication in 2016. It shows that approximately 87.5% of the people of the world are relying on their lives for the medicinal for the healthcare management using the medicinal plants at least at a times in their lifetime. So why the medicinal plants are so popular in the world? Because there are a lot of reasons, but however, we can say that briefly, yes, very easily available in your local territory. They have low cost of, or sometimes they are completely freely available in around our home sites, and they have very less side effect. And of course, they work synergistically because of the large number of the secondary metabolites present in the medicinal plants. And another main reason is that the medicinal plants are using by the our ancestors since the thousands of years ago, so they are already tested in the humans, so there is a very less chance of the side effect. Therefore the medicinal plants are being very popular nowadays. If we talk about the Himalayan medicinal plants, and particularly in the Nepal, we can see that Nepal is the beautiful country, very small country. It is located in between the India and China. So its area is just very small, 147,000 square kilometer, and population is the 28 million population just living in the country. So more than 10,000 plants are found in the Nepal. Out of them, approximately 10% of the medicinal, 10% of the plants are the medicinal plants available in the country out of the total biodiversity of the country. So another important point is that one third of the all species in the wider Himalaya range occurs within the Nepal. This is the another richness of the biodiversity. So 31st richest in the world in case of the biodiversity, Nepal. So if we see the richness of the medicinal plants of the country, there is approximately 20% of the flora are the medicinal and aromatic plants. Out of them, 1641 are the native medicinal plants found within the country and found only in the Nepal. It started originated from the Nepal. The native plants are the 1600 in the country. Similarly, if you see the potentiality of the medicinal plants of Nepal, so why today we are discussing about the ethnobotany and what is the potential of the country, I would like to share with this beautiful participants and in this beautiful event. So approximately 24 million US dollar every year, approximately this much amount of the economic export is currently being exporting from the country, that is based on the medicinal plants. So out of them, some of the medicinal plants like Sapandis or Bezinia or Xanthoxylum, these are the medicinal plants which are mainly exported in the foreign countries. So these are some plants which are mainly exported. And how the medicinal plants are being exported is that some of the medicinal plants are distill and prepare their essential oil or prepare their extract, but majority of the fraction of the medicinal plants are being exported in the crude form, in the raw form. They are exported in the other countries. So the picture shows that the utilization of this raw material of these medicinal plants within the country is very less, approximately 15% only. Out of the total production of approximately 85% of the medicinal plants are being exported in the foreign countries, either in the raw form or in the distilled or extracted forms. This is the picture. If we check the potentiality of the country on the basis of the plant resources, we can see that several pharmaceutical ingredients can be harvested, can be collected, can be isolated from the country, which are already in the mainstream medicine. Like you can see the morphine is there, curious, like quinine is there, curcumin is there, and ephedrine is there. Several bioactive compound can be exported by using the Nepalese medicinal plant. This is the potentiality of the country. If we talk about now I'm entering into my original topic, the traditional medicine of the Nepalese Himalaya, what is going on here. In the country, there are 4 types of the practitioners using the medicinal plants to treat the primary health care of the people, either in the urban area or in the rural area. The herbal medicinal based treatment is still in the practice. So there are diviners and spirituals group. They are utilizing the medicinal plants. There are herbalists, another group, they are utilizing the medicinal plants for the healthcare management. There are faith healers also using the medicinal plants, and there are traditional birth attendants. They are also using the medicinal plants, either way, for the help of the primary healthcare management of the local people. If we talk about the richness of the country, uh, Nepal is a very small country. It has approximately 10% medicinal plants within the country of total flora. 300, yeah, 300 diseases are currently being treated by using the local medicinal resources, particularly herbal plants. So there are several codified documents, there are several knowledges, approximately 4,500 manuscripts are there as a codified documents of the country. So herbal medicine practice is a very, it's a historical practice of the country for the primary healthcare management. Till the 1816, the database shows that we didn't had any allopathic modern medicine facility in the country. And before that, there was a practice of herbal medicine in the country. So we can see that several documentations are there. Several herbal practitioners are there. Several method of the traditional purification systems are there. This picture shows that the Silajit purification by the local healers. And there is the recording system, manuscript recording system. It's approximately 250 years ago. There was a recording system in the country started by handwritten documentation systems. They prepared the documents. If you check the formulation practice of these traditional healers, majority of the formulation practitioners are based on the Ayurveda. Though they are the traditional healers, but their root of the practice shows that they are practicing the Ayurveda. And another group of the practitioners shows that some of the groups particularly in the Himalayan region, in the high altitude, very high altitude, remote area, they are practicing the Chinese medicine and Tibetan medicine systems. And they are called the Amchi as well. And another group of the practitioners in the country are the Unani system of the practice. And similarly, homeopathic system is also being practiced in the country. So altogether, there are several traditional medication systems are practicing by the local healers as well as the mainstream. So you can see that in the picture that they are the Jharpuk, traditional practitioner, different type of the different tribes of the different principle-based practitioners are there currently practicing in the country. If you see the high altitude medicine systems, where there is lack of the transportation, there is lack of the modern medicine system, there is a lack of hospitals and modern medical facilities, the people are still relying on the herbal medicine, traditional medicine for their healthcare management, total healthcare management, not only the primary healthcare management. So their traditional knowledge is found that they pass down orally, mainly from their families. Their knowledge is passing down by families, and families, a series of families, they are passing their knowledges for the healthcare management of the people. So you can see this picture. This is the 3,600-meter altitude one practitioners is there. During our survey, we got this information. that 48 years he is a son of this is the 73 years old father. He is his father. So in this way they are passing their knowledge orally, mainly from one generation to generation and generations, but this knowledge is remain within their families. And this is the example of the remote area, that is 3600-meter altitude from the sea level, remote area of the Nepalese Himalaya. So this type of practice are going on in the Nepal. So, but they are particularly practicing the medicinal plant-based treatment system. Another system, it shows that the more than 2300 plant species are currently being utilized for the healthcare management. In Nepal, 125 caste of ethnic communities are there. They are speaking the several languages, but ultimately all the ethnic groups, all language speaking peoples are directly indirectly utilizing the herbal medicinal plants for their healthcare management. This is the example of this one district of the country. They are using the herbal medicine and they are formulating themselves. This is the traditional formulation systems. When the patient visit to their clinic, their traditional clinic, they just diagnose the problems, and then they formulate their own formulation and prescribe and ultimately give to the patient. So ultimately based on the traditional medicine system. This is particularly based on the Ayurveda. Another example, you can see that Amchi. It is the Amchi practitioners are based on the Himalayan region, high altitude regions. So they are. This photograph is taken during their workshop. So they are learning. They are learning how to formulate the product, how to diagnose the patient, how to prescribe, and how to give the medicine to them. And this Amchis are basically influenced by the Tibetan system of medicine, as well as the slightly influenced by the Ayurvedic system of medicine. Another example, you can see that this is the Bompo Tibetan Noble Monastery located in the Kathmandu. This monastery is the school of the Bonpo system of the Himalayan medicine. This is the school run by the monastery, and this monastery run the school for the medication system using the medicinal plant, but principle is based on their traditional Tibetan systems, not based on the Ayurvedic system. It is based on the Tibetan system. So these type of practitioners are available in the country. If we see the ethnomedicinal practices, if we talk about the Ayurvedic practice in the Nepal, the Ayurvedic practice in Nepal is, of course, it is started from our ancestors since 5,000 years ago, from our generation to generations. But the recording system is started from the period of the Chandra Samset, that is called about 250 years ago in the Nepal. The written manuscript is recording system is started from at that period it was completely handwritten, and this sketch is completely made by the hand, and the photograph of the plant is sketched by using the local resources, local colors, natural colors, and completely handmade. And the description is written in this way. So this is called the Chandra Nigantu. There are several Nigantus in the Ayurveda. Out of them, this is the Chandra Nigantu record, local Nigantu of the Nepal, recorded some 250. So this record comprises 940 medicinal plants, and as well as 336 minerals and animal products in this Nigantu. So that was completely based on the Ayurveda. Another herbal practice, if we see in the Nepal, this is the researcher while doing field research, he get fractured his leg, and this traditional practitioner giving some medicine for the treatment. So the traditional practitioners collect the medicinal plant and process for their local, based on their local practice, and they prepare some medicine by different traditional medicine formulation system. This is the preparation of the drug, and then ultimately they prepare the bhatti, and then they diagnose themselves according to their own system. And this is the herbal practitioners in the Nepalese Himalaya. They are practicing medicinal plants. But uh, ultimately, all these practices are mostly based on the Ayurveda. It is influenced with the Ayurveda. If we talk about the ethnobotanical knowledge, what is going on in the Nepalese Himalaya is it shows that most of the cases, most of the people, most of the illnesses, most of the practitioners, they are practicing for the gastrointestinal elements. That is the disease-wise. If we see the gastrointestinal elements is the most of the medicinal plants are the prescribing for the gastrointestinal elements. So most of the medicinal plants are collected from the wild habitats, not the cultivated one. If you see the another data, it shows that on the basis of dosage form, most of the cases, the juice dosage form is the highly used dosage form, highly used practice in the Nepalese Himalaya. If you see the plant part wise, the underground part is the highest utilized part of the medicinal plant of the Nepalese Himalaya. It shows that altogether, it shows that underground part in the juice form, particularly for the gastrointestinal treatment, is the highest practice in the Nepalese Himalaya. This is the information published in 2013 and 14. And then, yeah, another research again shows that yes, the highest in the gastrointestinal elements, the herbal practitioners are using the herbs for gastrointestinal problems. Let's talk about, this is all about the introduction of the Nepalese ethnobotany and ethnobiological practices going in the Nepalese Himalaya. I would like to briefly introduce about what is the research going on about our laboratory, our teamwork doing on these Nepalese Himalayan medicinal plants. Particularly, we collect the medicinal plant on the ethnobotanical guided based and as well as the random based. Mostly we collect the random based. We visit the site and collect the random based and bring to the laboratory. As well as also we talk with the local people and ethnobotanical best and bring to the laboratory and do some different type of the research activities. Some examples are today I'm presenting here. So this is the example of the mango peel collected from the local area. And then finally, we carried out one research. In this research, mango peel shows that the cell viability, this is the anti-cancer activity, cell viability assay in HeLa cell viability was reduced up to the into 200 micromole doses form. And finally, but the mango flesh did not show such type of result. In comparison to quercetin, a similar result was obtained from the mango peel. Mango peel and quercetin both able to inhibit the cell viability in case of the HeLa cells. But mango flesh, which we usually eat as a fruit, did not show such type of result. And ultimately, we concluded that the the HeLa cell, through cell induced the cell death by the apoptosis because of some of these reasons. One is that the increase the cell population in the sub G1 phase. Another reason is that appearance of the fragmented nuclei. You can see in this figure, this is appearance of the fragmented nuclei. And another reason is the changes in the distribution of the nuclear chromatin. I will show in the next figure changes in the distribution of the nuclear chromatin. So all because of these reasons, it shows that there must be the apoptosis. Another reason is that the mango peel inhibited the cell viability by the ultimately expression of the caspase 7 and caspase activation, caspase 9, both activation. This could be the one reason, and this is the fragmented nuclei, which shows that thus the extract induced the apoptosis during the cell division or cell culture. Similar type of the research we conducted in the orange peel as well. This is the mango peel, and we analyzed the chemistry. We analyzed the chemistry of the mango peel, and it shows that both GCMS and LCMS analysis shows that there are some of the phytochemicals which could be responsible for the inhibition of the cell viability, like kerosene. Some phenolic compounds are there, and some unsaturated fatty acids are also there. These major phytochemicals could be the responsible for the inhibition of the cell viability. And similar research we conducted in the orange peel, and again the orange peel again shows that cell viability percentage is reduced up to 400 microgram per mole in case of the peel. But in case of the HeLa cell, if we see about the AGS cell, also similar result was reflected. If you see the chromatin, again similar result was reflected. So cell line revealed close condensed bright and blue nuclei in peel extract treated cells that may be the reason of the apoptosis. This is another conclusion, and we analyze the chemistry of this this orange peel, and then chemistry shows that beta caryophyllene and other compounds are the major chemical compound which are present in the extract of the orange peel. The beta caryophyllene and aroptine could be the member, could be the leading member for the inhibition of the cell viability. We concluded like this, and then later on another group, some publication I have found that yes, beta caryophyllene inhibits the cell viability. Some another research carried out in this compound reflected the similar result. Similarly, another group carried out the another research in the aroptine. Aroptine shows that yes, this is the aroptine is responsible for the apoptosis. So similar research we again carried out in another plant in the leukemia. So we prepared the extract, and then we first determined the antioxidant activity by the DPPS assay. It shows the very nice antioxidant activity in comparison to the ascorbic acid. And then we tested in the leukemia cell viability when we compare up to the 200 microgram per mole. It shows that the very strong cell viability, cell inhibition of the cell viability. So it concluded that yes, Euphorbia hirata could be the another member for the anti-cancer activity. So this is the another plant. And similarly, we again carried out the similar research. Yeah, in the another two plants like Dipsacus mitis and Woodfordia fruticosa, we conducted the cell. This experiment was carried out in the smooth uterine smooth muscle cell of the mice, and then it shows that up to certain dose, it shows that the completely contraction in this person, is the contraction relaxation going on up to certain time interval. It shows the completely contraction of the uterine smooth muscle cell after the treatment of plant extract. So it shows that the effective weight of 30,000 microgram in the tissue, 50 gram of tissue is showing that the contraction of the smooth muscle cell of the non-pregnant rat. So why we taken this plant is that this Woodfordia fruticosa is traditionally used in Nepalese Himalaya for the management of the gynecological disorders. So this type of research justify that why the people were using this plant for the gynecological disorders. Because contracted cell facilitate the journey and expels particularly endometrium during the menstruation. This experiment justify such type of the conclusion. Similarly, we another on the basis of traditional knowledge, the traditional knowledge, it was 2016, Paudel and Singh shows that the calotropis is used for the inflammation and cramps, and we bring this plant to our laboratory, and then brought to the laboratory, and we evaluated the anti-inflammatory and analgesic properties. If we see the analgesic effect of the Calotropis procera, up to the 62.5 milligram per kg body weight, 70% inhibition was obtained in comparison to the aspirin. Aspirin obtained the 93% of inhibition of the analgesic activity. It was carried out by acetic acid withering mice assay. Similarly, if we see the anti-inflammatory activity in the paw edema assay, calotropis in the same dose, it shows that 29% inhibition of the paw edema, formalin-induced mice paw edema. It was compared with aspirin. So on the basis of traditional knowledge, people were using this plant for the using the calotropis for the management of analgesic and anti-inflammatory properties. We brought to laboratory and justify the scientifically for the similar type of the research. Yeah. Another experiment we conducted in the essential oils. Normally, the essential oil, like one essential oil, rhododendron anthopogone, is used in the skin healthcare management traditionally. It is according to the Siva Koti. And we brought this plant in the laboratory and evaluated for the antimicrobial property, which are related for the infection or problem causing in the skin, particularly Streptococcus aureus. Streptococcus aureus, it causes the illnesses, minor illnesses in the skin. So when we tested this plant, rhododendron anthopogone oil, in the laboratory, particularly, it shows the inhibition of the zone of inhibition, that is inhibition of the microbial growth during the laboratory tested. What we again added in this is that we loaded one essential oil in the one side and again loaded in the standard antibiotic in the another side. So the synergistic effect of the rhododendron oil and erythromycin on the Staphylococcus aureus shows that there is a synergistic effect, and in this concavity, inhibition of the microbial growth is clearly seen. That means if we we can able to utilize both legally and practically essential oil and the standard antibiotics together in this pattern, it could be more beneficial to reduce the load of antibiotic in the human consumption. Another research we conducted that particularly these are some medicinal plants. There are 12 medicinal plants. We first surveyed the total phenolic content in the plant, and then again we surveyed the antioxidant activities of this plant. The reason behind that was we were searching the old healing herbs from the Nepalese Himalaya, but we traditionally know that these medicinal plants are using for the own healing properties by traditional practitioners. So we brought them in the laboratory. We evaluated the total phenolic content. The plant which were rich in the phenolic content, assume that tannin, the plant which were rich in the phenolic content, we selected only those, and then we again forwarded for the antioxidant activity. From them, we able to conclude the 5 medicinal plant having the potential antioxidant activity. Out of 5 medicinal plants, we chosen just top 3 medicinal plants which were strong in the both total phenolic content as well as strong in the antioxidant activity. Just we selected the 3 medicinal plants: Myrica esculenta, Boinia variegata, and Rhododendron arboreum. So ultimately, we formulated them in the ointment. We formulated them in the ointment in the ratio of the 1:1:2 ratio in the ointment form. And then finally, we tested with the animal, because traditionally we know that they are used for the wound healing property. There is one again literature is in 2012 shows that yes, the Myrica esculenta is used for the wound healing property. So this is the reason why we chosen this plant. And then while we tested this plant for the wound healing property in the animal, up to 9 days result, we found that the ointment prepared from the crude drug, there is a 0% of the wound remaining in the animal, in the mice, 0% remaining. While pharmacine was the standard. There is a 2% is still remaining in the pharmacine treated group. One was the control and another was the blank. It shows that the traditional practice is justified scientifically, that yes, it is working for the wound healing property. And similar research work we conducted with the Diploma butyracea. This plant is also traditionally yes used for the cracked heel, lips, skincare management in 2006. This is the reference. This is also used for the emollient for chapped hand and the feet in the winter. This is the reference of 2012. So what we did, we evaluated the anti-inflammatory property, and then we again evaluated the analgesic property, and again we evaluated the wound closure response, because traditionally it was known for the cracked heel, lips, skincare management, and chopped hands, etc. So results shows that in anti-inflammatory property, percentage of inhibition of the inflammation was diplopnema oil. This is the seed we obtained. We extracted the butter from this seed. Diplopnema oil or butter is equally able to show the result anti-inflammatory property in comparison to the diclofenac. So similarly, in case of the analgesic, also the similar result it is comparable result with the diclofenac. Similarly, in case of the wound healing property, it is also comparable result with the pharmacine. So it is shows comparative results. So it is altogether indicating that the traditional practice of the Nepalese Himalayan medicinal plant is scientifically justified. And another example of research we conducted, that this is the herbal powder formulation. We collected the anti-diabetic herbs which are currently in the practice, which are edible, which are currently we are using in our kitchen, as well as the local practitioners are prescribing their patients. There is no risk of the side effect, toxicity, or any danger. So we selected total 12 medicinal plants for the anti-diabetic properties, and we randomly formulated in the 80%, 10%, and 10% ratio. And from this result, formulation number 7 shows that prominent result. This is the diabetic volunteer. It has the initial sugar level is 213. After the 6 hour, the diabetic patient reduced its sugar level significantly in 100, just drastically reduce the blood glucose level. Similar research we conducted that in case of the anti-cancer activities, we chosen the breast cancer cells, and there are some other medicinal plants like Hordeum vulgare, pomegranate, and green tea extract we prepare, and Hordeum vulgare shows the significant anti-cancer activity with the breast cancer cell. We continued this research. Previously it was 3 medicinal plants, and later on we collected the 8 medicinal plants for the anti-cancer activity, and we evaluated the antioxidant activity, and all together shows the very strong antioxidant activity. While we mixed these medicinal plants synergistically together and prepared one formulation, that formulation was the mixture, and this mixture shows the very strong antioxidant activity, that is IC50 value is just 17.5 after the mixing. Individually, they were very high IC50. After the mixing, synergistically 17.5. And then what we did, we prepared the capsule from this and evaluated the different parameter of this capsule. And again, we sent this sample to the foreign laboratory for the evaluation of the anti-cancer activity. It shows that, yes, there is the significant inhibition of the cell viability by using this mixture for the control of the breast cancer cell as well as cervical cancer cell lines. All this information was based on the traditional practices collected from the Nepalese territory. And probably this is the last one. We formulated the polyherbal cream by using the 3 Glycyrrhiza and Azadirachta indica and Cinnamon gelanicum extract in the different ratio. After then, what we did, we prepared one cream. And first of all, we optimize the formulation base. For the formulation base, so we stearic acid and cetyl alcohol. We two main the fatty acid utilized in the formulation, and we fix the relationship between the response and independent variables between these two fatty acid, taking the viscosity, pH, and spiralability as a base. After then, we evaluated the formulation with the formulation ratio number 3. Ratio number 3 means is given here. Different ratio was a very good antioxidant activity by using both DPPH and lipid peroxidation assay. Formulation number 3 was excellent. So this formulation number 3, this is the formulation number 3 in different ratio, and the previously confirmed base. We mixed this base and prepared the one ointment. And while we prepare one cream, while we evaluated this cream for the permeation profile, it shows that the cream followed the first order of the permeation profile, and thus the stability and the pH, viscosity, spiralability, irritacy test all were good after the evaluation, up to 3 months. Similar type of research we carried out in the wild allium and the cultivated allium species. A very strong antimicrobial activity was shown by the allium species. Another research we conducted in the this Astelvis rabicularis, Chulokati it is called, Astelvis rabicularis. It is traditionally known for the very good herbs to control the diarrhea. Therefore, we brought to the laboratory and tested for the antimicrobial activity, particularly against the E. coli. In justification of the traditional medicine, it shows that a very good inhibition with the E. coli. It shows the 13 mm inhibition, zone of inhibition with the E. coli. So since traditionally it was used for the diarrhea, this type of research justify the traditional application. And finally, we formulated into the tablet form. Conclusion is that the botanical based products are backbone of the healthcare management in the Nepal. Traditional knowledge of the Nepalese practitioners are passed down orally, mainly within the families. They are not happy to share this knowledge with the other people. The pharmacological evaluation of the Nepalese medicinal plants shows wide range of activity having the possibility of herbal formulation and bioprospecting. With this, I would like to acknowledge all these institutions which support this research, and my students which carried out all these research activities, and particularly special thanks to the SMBT Sevabi Trust, SMBT Institute of Diploma Pharmacy, Professor Yogesh, and organizing team of the second international conference on traditional medicines. And thank you so much.
Thank you so much, sir, for the nice presentation. I request participants, if you have any question, just put it in chat box. If anyone wants to ask anything, you can ask. The session is open to all. I think we don't have any question from the participant. Yes, sir, we are having one question. I request Dr. Mutha sir, please go ahead and ask your question. He's with us. Please.
Good morning, sir.
Hello, namaste.
Sir, in last part of your presentation, you have explained the things like these traditional healers are refused to tell their knowledge to others. just when we visit to such type of places, like in our region there is Toranmal. We just used to visit that particular place many more times, and we just discuss the things with the traditional healers about the practice they follow, about the plant they use for various ailments. But what strategies that has to be followed so as to document these things so that it will be permanently useful for the next generations? What is your opinion?
Thank you, sir. Normally, their traditional knowledge is the only the way of their income. So they are not happy to give or transfer this knowledge with others. But to get their knowledge preserved for the generation to generation as a property, I think government or authorities should pay some huge compensation or facilities or bring them to some special environment so that they make sure their employment. Otherwise, they are not ready to hand over their secret. So that could be the one strategy, that if we government or authority can facilitate them for their education, living, hand to mouth, and other all facilities, in that situation probably they will be ready to transfer their knowledge. Otherwise, it is very difficult here in Nepal also. Superficial knowledge they are giving us during the survey, but in-depth knowledge they are never transferring, even our volunteers as well. So strategy should be taken by the government.
Hello.
Yeah, please.
Sir, can we make any tie-ups with these persons whenever we reach to some certain formulation by using their knowledge? Then we can make a tie-up with them, and whatever the financial benefits we are getting, we can share with them. So that may be the one strategy that we can use so as to make them speak and document the things they are with from last many more years.
Yeah, I agree with that. Benefit of share in the traditional knowledge, NOEGA protocol, as we say that NOEGA protocol, that could be also implemented in such cases. Yes, if we share the benefit, but it's still very difficult to collect their original knowledge in that. Yeah. Okay. Agree with you.
Thank you, sir.
Thank you. Thank you.
Thank you so much, sir. Again, I'm asking participants, if you have any questions, then just ask as early as possible. I think we don't have any question from the participants. Moving further, I would like to thank Professor Dr. Gavli for putting some time for us, for reserving time for us from his busy schedule. And apart from all these things, we would like to thank you. We would like to thank you for accepting our invitation, and we are looking forward for your physical presence on the next year for third International Conference on Traditional Medicine 2022. Thank you very much, sir. Your work was really, really nice. We admire your students also, who is a part of your research work. I think we have one question, right? Yes, program chair, Dr. Yogesh Mishra, asked you one question. Yes, sir. Dr. Go ahead.
No, sir, it's not actually question.
Okay.
Please, sir, please.
Okay.
No, not a question. It's just I want to say thanks from my chair. I'm very thankful to you spare some time for my second ICTM. But I just want one assurance from your side. Next year, physically, your presence for the third ICTM,
Yes, sure.
That I am requesting from my organizing committee.
Thank you very much, sir. Okay, definitely I will be there.
You
It's
wonderful
my pleasure.
speech.
My pleasure. My pleasure.
Yes, thank you. Thank you.
Namaste, sir. Thank you very much.
Namaste.
Thank you so much, sir. Thank you, Dr. Ushir, and thank you, Dr. Gyavli, for putting lights
Thank you.
on the path of young research. It will be always a guiding path for them. Again, I am thankful to Professor Gyavli on behalf of SMBT Educational Trust and the entire organizing committee of second ICTM 2021. Thank you so much.
Thank you. Thank you so much for SMBT family, entire family. Thank you so much. Thank you giving me this platform. Thank you very much. Okay. I'm leaving this. Okay. Thank you.
Yes, sir, yes, please.
Please. Bye.
And the about next session, it will be sharp start at 12 p.m. Okay, till then, please stay connected. We'll be having a last session which will start at 12 noon. Thank you so much.
Good morning, sir. Good morning, everyone. I am Bhavna Tambe, Professor SMBT Institute of Diploma Pharmacy, Dhamalgaon, Nashik. Uh, Good morning, everyone. It's my pleasure to introduce our today's speaker, Dr. Sherry Patunde Ademi, sir. Sir was completed PhD from UCA Tarsadea University, Bardoli, Gujarat State, India in biotechnology. Sir completed MSc Ethanobotany in 2011 from University of Ibadan, Nigeria. Sir has completed BSc in Plant Biology in 2007 from University of Ilorin, Nigeria. Sir has many more experience. In 2018, Sir worked as a lecturer in Department of Plant Biology, University of Ilorin, Nigeria. In 2015 to 2018, Sir worked as a lecturer in Department of Plant Biology, University of Ilorin, Nigeria. In 2012 to 2015, Sir worked as an assistant lecturer in Department of Plant Biology, University of Ilorin, Nigeria. Then Sir also worked as a honor distinctions and membership of learned societies. Sir also worked as a member society of the Conversation of Phytophil and Science Sense in 2013. In 2013, Sir also works as a member Nigerian Society of Experimental Biology. In 2012, Sir worked as a member Society for Economic Botany. In 2012, Sir also worked as a member Botanical Society of Nigeria. And in 2012, Sir worked as a member West African Network of Natural Product Research Scientists. More than 30 publications and papers was published. Now I request you, sir, please continue your session.
Good morning.
Good morning.
Yeah, I'm Adeyemi Sherry Babatunde from Nigeria presently. My PhD biotechnology in Ukatasa is still in view, it's not yet completed. Just to correct the citation. I would like to state that I belong to two institutions presently, University of Ilorin and Ukatasada University in Badoli, Gujarat State, India. But presently I'm on leave in Nigeria, that's why I'm in Nigeria. So I would like to speak on herbal medicine for diabetic mellitus treatment, status and prospects. Before we continue, we should know the distinction between traditional medicine and herbal medicine. Traditional medicine refers to health practices, approaches, beliefs that incorporate several things. Aside plants, they can incorporate animals, several things, even some kind of dancing and music is part of treatment. Void of conventional use of drugs is traditional medicine. And according to WHO, is the knowledge, skills, and practices based on the theories, beliefs, and experience of indigenous to different culture. So this means that in the culture, they may have different beliefs about treatment of diseases and ailments, of which another culture, they may say, okay, only water is treatment, and you have to admit that as their traditional medicine. Some people may say, when they are sick, they dance. And this dancing is a therapy towards alleviating their disease conditions. And herbal medicine is herbal medicine are naturally occurring plant-derived substances with minimal or no industrial processing that have been used to treat illness within local or regional healing practices. And herbal medicine, from the name, you will agree is you source it from herbs. And these herbs are nature's gifts, which are found virtually everywhere. Even Antarctica, where you have seaweeds and very cold condition, you have plants there. And these plants have been put there to serve purposes. Purposes of disease treatment and alleviation of disease conditions. And there is no way we can start isolation of compounds or discovering compound without talking about the plant knowledge. The healing processes from early man started from usage of plants, because there was no conventional treatment then. So in my own view, the oldest form of treatment is herbal medicine. Medicinal plants have several potentials. They have been used to treat malaria. They have been used to treat or manage sickle cell. They have also been used to manage fertility and have served as abortifacient in case of problematic pregnancy. They have used plants to abort pregnancy if the pregnant is having problem. Also, they have been used as anti-neurodegenerative and many more. And more recently, this COVID-19, so many researchers have come out to state the importance of using medicinal plants to alleviate this condition. Because we believe that medicinal plants have little or no side effects if used as prescribed. Yeah, diabetes mellitus, which is the main subject matter here, is a group of metabolic disease conditions which is characterized by hyperglycemia, because there is a defect in secretion of insulin, there is defect in action, or both. That's what takes us to have like 2 types or 3 types, at times 4 types of diabetes. But we are very popular with 2 types, type 1 and type 2 diabetes. When your blood sugar is raised, this is a common effect of uncontrolled diabetes. And over time, there is the severe damage of several body systems, the nerves, and the blood vessels. If you have diabetes, there is 100% assurance that these cells and vessels will be damaged, and which will affect different organs in the body, the eyes, the kidney, the nerve, the heart, and the blood vessels. So these are the types of diabetes commonly known, type 1 and type 2. Type 1 diabetes is as a result of problem in the pancreas. There will be shortage of insulin production from the pancreas, and if there is shortage of insulin production, there will be excess blood sugar there will be excess sugar in the blood. So that's why it's insulin dependent diabetes. While type 2 is non-insulin dependent. There's production of insulin, but there's something with the absorption of glucose from the blood into the cells. So these are the 2 diabetes we'll be talking about, and for the sake of this presentation, I'll be calling them diabetes or DM. So these are the chronic complications of diabetes according to Diabetes Atlas. So there is diabetic eye disease. We have several other complications of diabetes. And symptoms of diabetes, one will be always thirsty, always tired. There will be sexual problem, wounds that won't heal. frequent urination, hunger, and systemic weight loss. So we know diabetes is found throughout the world, but how prevalent are they? But thanks to researchers that have really worked there to tell us and to project for us, if care is not taken, the number of diabetic patients what we experience in the nearest future. Presently in the world, in 2019, there were 463 million diabetic cases, which was projected to increase to 578 million in 2030 and 700 million in 2045. If care is not taken, we may have more than this. This is just a projection. If we don't take care of our lifestyle, we may have more than this. And in Africa, it's projected to increase by 143% by 2045. Also in Middle East and North Africa, 96%, and Southeast Asia, 74% increase. And if we don't look at the status of medicine, because they've been using the conventional therapies and patients are faced with several side effects. And we understand that medicinal plants have little or no side effects. So the best thing for the natural product scientists to do is to keep looking and keep searching for medicinal plants that will solve this problem. And I hope very soon we're going to solve this problem. Yeah, this page, this chart is just telling us about the prevalence by age and sex, and also the projection, which I've talked about, and also the top 10 countries for number of diabetes. The top 10 diabetes, the top 10 countries for number with diabetes. China is topping the list, and by 2045, China will still be at the top, while followed by India and Pakistan, Brazil, Mexico, Egypt. They are top 10. So meaning that if care is not taken, some countries may even overtake China and India, so to say. Because if you don't take care of our lifestyle, more cases will be springing up. So conventional management of diabetes and their limitations. Yeah, there are several classes of oral hypoglycemia or hypoglycemic drugs that have been used and that exerts antidiabetic effects through different mechanisms. For instance, oral sulfonylurea acts to reduce blood sugar mainly by elevating insulin release from islet of Langerhans. This mechanism only works with insulin, and if we use sulfonylurea and its derivatives, you have weight gain as side effects, and you have to be using insulin almost every time with the drug. And we have biguanides, which acts to reduce hepatic glucose and hepatic gluconeogenesis, and to replenish peripheral tissues which are sensitive to insulin, and these are achieved through elevation of insulin stimulation, uptake, and use of sugar. And it's also ineffective in the absence of insulin, and several other like that. So let's now talk about herbal medicine. We know the conventional therapies have been faced with several challenges of side effects. So how do we come in using medicinal plants, herbs to rescue the situation? Several people have written thousands of papers on usage of medicinal plants and their derivatives in the combat of diabetes mellitus disease. And I came up with these publications from PubMed to show the number of publications that have addressed the issue of diabetes from 1980 to 2000 and from 2000 to year 2020, December 31st, year 2020. And you can observe from this graphical presentation or illustration that Europe is still having the highest number of publications, followed by Asia, and followed by Africa, by others. Even in Antarctica, we have researches from there addressing diabetes mellitus treatment using seaweeds and several natural products there, and several other researches from ethnopharmacology, from Journal of Herbal Medicine. You see several research on medicinal plants usage to alleviate diabetes mellitus condition. And though this is made possible by the reported phytochemicals in medicinal plants. We know these medicinal plants contain secondary metabolites, which are products of plant metabolism that do not take part in the growth of medicinal plants. So meaning that these secondary metabolites have been produced for the benefit of man. So if we don't tap into them, yeah, we are wasting them. So you have to tap into medicinal plants to use the natural products therein and to see the benefits therein. I've come up with several collections of some plants that have been reported to either secrete insulin or to mimic activity of insulin. So we have Acacia arabica, Allium sativum, Egu mamelos. We have Agrimony, Annona squamosa, Camellia sinensis, Zingiber officinale, Hibiscus rosa sinensis, and several other like that. You have Aloe barbadensis, that we call aloe vera, Capsicum frutiscens, Bavia diffusa, Eucalyptus globulus, Ficus religiosa, Cinnamomum ceylanicum. We have several other plants, but this is just to show us several researchers have worked on medicinal plants and have reported their importance, and we have these plants around us. If we check out the problem we're having with this study, I'll still come back there. So we have also some plants that have been used for glucose uptake, that they act as glucose uptake agents, the same way you have in conventional therapies. We have Juniperus communis, Salix, we have Alnos, Costus pictus, Lycopodium, and several others like that. These plants have been reported to act as glucose uptake active agents in case there's problem in the glucose uptake. Kind of activity has been is similar to what biguanide does in the system. Also, we have alpha amylase inhibitors plants. We have Aloe barbadensis, which has been reported by Abu Soud et al. In 2004 to inhibit alpha amylase and at 80 microgram per ml. And we have Morus alba, which has been also reported to inhibit at 1,440 microgram per ml. Also, Eugenia communis seeds, which has been reported to inhibit alpha amylase at 632 microgram per ml. Also, we have alpha glucoside inhibitor plants. We have mucuna pruriens, which has been reported to inhibit alpha glucoside at 0.8 microgram per ml. This is very active. At 0.8 microgram per ml, there is alpha glucosidase inhibitors. And most researchers now, due to, especially in India, where you have ethical issues towards using animal for experiments, they use alpha glucosidase in vitro assay and alpha amylase in vitro assay to check the activity of the plants and their derivatives. This alpha glucosidase is one of the best assay to check the diabetic activity of plants and natural products. Also, we have camellia sinensis, which has been reported to be 299 microgram per ml active. This 0.8 and the rest of this number are their IC50, as reported by the researchers. Also, we've been able, researchers have been able to source phytochemicals from plants, and they've reported these phytochemicals and the targets of the phytochemicals have also been reported. For instance, catechin and apigallocatechin gallets have been reported to be isolated from Camellia sinensis and Theobroma cacao, which is cocoa. We have that in Africa. And these two compounds have been reported by the following author to be antioxidative. They have protective effects against oxidative damage. They modify oxidative stresses. They reduce lipid peroxidation by enhancing SOD, GST, and CAT activities. Also, from Cocoma longa, there was curcumin as active ingredient from there, and have been reported to lower blood glucose and also to lower glycosylated hemoglobin levels. Also, camphorol, which has been reported to be present in several plants, have been reported to promote insulin sensitivity and preserves pancreatic B cell mass against destruction. So this camphorol may be useful for those that are having type 1 diabetes, but type 1 diabetes prevalence is less than 10% throughout the world in all diabetes cases, whereby we have 90% plus for type 2 diabetes, which is the commonest and the most common diabetic cases. Also, we have other compound like embelin, which has been isolated from Embella ribes and Lysimachia punctata, and has been reported to reduce elevated plasma glucose, glycosylated hemoglobin, and pro-inflammatory mediators. And also erianin, which has been reported by Yu et al. 2016, and isolated from the Dendrobium cristotoxum, which has been reported to inhibit high glucose, induce retinal angiogenesis, fear blocking of different pathways. Also, we've had garcinol from Garcinia indica, which has been reported to decrease plasma insulin and glycogen, high density lipoprotein, and also it has been reported to act against some antioxidant enzyme, and it causes significant reduction in elevated level of blood glucose, glycosylated hemoglobin, and lipids. Also, we have been able to see researchers finding out how medicinal plants can also be checked in human in form of clinical trials, which we have in conventional therapies. And this kind of trial is usually done using the herb, not the compound, the herb and conventional therapy to see the synergistic effects between the two, so as to recommend plants to patients, as well as telling them the safety when they use with different oral hypoglycemia. And this research has been reported, this was done in 1996 by Bunyapra, I can't pronounce the name, it's very, very long. And this research had 72 type 2 diabetic patients, 49 men and 23 women aged between 35 and 70 years with high level of fasting blood glucose and a typical diabetic cough of glucose tolerance analysis. At the end of the day, this patient did not show response to glibenclamide alone, while the aloe vera juice significantly reduced the level of fasting blood glucose within 2 weeks and was reported to be safe for both the kidney and the liver. So meaning that aloe vera juice was tested against patients having type 2 diabetes, and it was found to be safe. And it was found to reduce the fasting blood sugar, blood glucose within 2 weeks of usage. Also, aloe vera complex gel has also been reported to be used against 136 patients with prediabetes and type 2 diabetes, and which are not on any medication. And their information as reported here. This clinical, this trial showed borderline significant reduction in body weight, body mass fats, fasting blood glucose, fasting serum, insulin, and IRA after 8 weeks of treatment. So meaning that the claim of indigenous people for using this kind of therapy is upheld, and they are very correct for using medicinal plants to treat their disease conditions, including diabetes. Also, Juglans regia was also reported and has also gone through human evidence trial, and it was found to improve blood glucose by reducing the FBG, the glycosylated hemoglobin, and several other parameters in the system as compared to placebo treatment, which did not affect the liver and the kidney, but rather showed gastrointestinal disorder, which can be managed. But the aim of using this treatment is already achieved for reducing blood sugar, which is one of the leading causes of death in the world. So we have other researchers also that have reported clinical trials on different plants. Tinospora cordifolia as reported in 2007 and 2017, Sauropus androgynous in 2002, 2015, and 2016. Gingiva officinale also reported in 2013 and 2014. These are clinical trials. This kind of trials, the requirement is different from the conventional therapy clinical trial, because one of the challenges of usage of herbal medicine is the clinical trial. But WHO came up with a regulation which is solely for medicinal plants clinical trial. This has helped a lot of researchers to check their herbal medicine uh, uh, using human evidence. We know that using rats will give us some insights towards the usage and the efficacy of the compound. But the physiology of man and rats are different. We may just have an idea that, okay, this is this, but when it comes to clinical trial or human evidence, the result may change. And we cannot rely solely on in vitro evaluation of our drugs. So we should also be able to use human evidence, especially when we know the drug we are giving is non-toxic. So that's one of the problem, toxicity. So what's the prospect of using herbal medicine for diabetes treatment? Yeah, we know usage of herbal medicines, medicinal plants were verbally passed down to generations. But a few years back, the WHO encouraged researchers to document the usage or the reports from the local people, which are very close to culture. We should go to them in form of ethnobotanical survey, ask them the plants they found around them, what the plants are used to treat, like that, how the plants are used, so that they will not die with the information. Because if, let me give instance of artemisinin, which is a product of plants from Artemisia annua, which is a precursor of several anti-malarial drugs in the world today. This artemisinin was isolated from Artemisia annua. For instance, if this information about its usage has died with the local people, will there be artemisinin today? No. But thank God the information was still alive and they could get artemisinin from Artemisia annua. Likewise, we, natural product researchers, need to visit the local people and ask them what plants are used to treat diabetes mellitus in their locality and how these plants are used. The prescription. Also, the prospects, the use of technology and is helping this research or this the research of finding solution to diabetes mellitus, because there are a lot of technology now to find out whether your plant is active or not. And using technology, a lot of mystery, a lot of myths will be unraveled. And there won't be, okay, when you collect these plants in the night, it may not work. Yeah, we can use technology to find out. If when I collect the plant in the night, in the afternoon, we can check the activity of the collection time also. Also, the prospects, number 3, which is stated here, is it's an important contribution to economy, because the demand for herbs is on the increase, owing to the fact that they are very active, they are cheap, they have little or no side effect. And several pharmaceutical companies now rely on products from medicinal plants to alleviate disease condition. So the demand for herbs on the increase, which is going to add to the GDP of any country, especially in India and China, they are the leading exporter of medicinal plants, and very soon other countries also will join, because it's not eye opener that we cannot do without using herbs. And for natural product scientists, you can also key into this by growing medicinal plants or sourcing for them, and from this we get we may be gainfully employed using exporting medicinal herbs to some countries of needs. Also, we have challenges of developing herbal drugs for diabetes mellitus. I've thought about the clinical trials. It was once a challenge, but now we are getting over it, because the WHO has given regulation towards testing herbal medicines in clinical trial. Also, analysis and documentation of toxicological and epidemiological and other pharmacognostic-based data and verification of herbal materials is also a challenge. To reduce adulteration, we have to verify the herbs, and also to document toxicology is also a challenge, which several researchers are now trying to solve the problem. And also, evaluation of drug interactions is also a challenge, and standardization is also a challenge. All these challenges are being worked on, and very soon I hope we get over these challenges. And also, therapeutic actions depend fundamentally on age, location, and part of the plant's species used. For instance, I may say from Nigeria, a particular plant is used for diabetes at this pace. Maybe I give the IC50, but the same plant may not be active if sourced from Indonesia or sourced from India, because most of the time, since this plant, these are the constituents are secondary metabolites. So this secondary metabolites varies based on the age of the plant, the time of collection, the age of collection, the location of collection. So this is also a challenge, because if I say a plant from here, maybe a plant from India, for instance, let's say Gymnema sylvestre from India, which is also known as anti-diabetic plant. If I find the plant in Nigeria, it may not be as active as the one found in India because of the continental drift, the geographical location, and also the challenge is timing of harvests, and adulteration with microorganisms also is a challenge, and which may affect the standard of herbal medicine globally. So, conclusion: for centuries, almost every known human civilization has been using medicinal herbs as effective medication for prevention and treatment of multiple health conditions, including diabetes. This is because of the general belief that herbs are without side effects. They are cheap and they are easily accessible. And recent introduction of cutting-edge analytical techniques and research methodologies, which has increased evaluation and validation of herbal medicine, will make herbs to be continually explored for their efficacy. Diabetes mellitus, as many other ailments, has been reported to be managed using herbs and their derivatives. It is foreseen that herbal medicine will be integrated into conventional medical system, as the herbs are nature's gift. So before I say thank you, this is my university in Nigeria, which was founded in 1975. Is the first choice institution for admission seeker, and these are dam for recreational purpose. This is just the area view of part of the college. Our college is one of the biggest in Nigeria, with top-notch research facilities and hostel facilities for students. Thank you for listening. This is my information down here. And also, I'm open for collaborations. Thank you.
Good afternoon, Professor.
Good afternoon, sir.
Okay. We have a request from program chair first, Dr. Ushir. Do you want to say anything? Okay, I request participant, if you have any kind of question, then put it in chat box. Anyone have any question from the participant? I think nobody have any question. Lecture was so good, sir. Thank you so much for sharing your lecture with us. I would like to request Dr. Yogesh Ushir, Program Chair of Second International Conference on Traditional Medicine 2021. Please share your views regarding this presentation.
Good morning, doctor.
Good morning, sir.
Okay, so it's a good afternoon in India. So namaste
Hi.
from my whole entire team. Namaste from India. So
Namaste from Nigeria.
very nice to see you. So
Yeah, thank you.
very nice interaction with you, and I like to give this special thanks to Dr. Admei, who pick up the chance to deliver the lectures to the audience from the India and even the overseas audience. So thank you very much, Dr. Admei. One question is in my mind. You just share the views regarding the diabetes medicinal plant. There is any formulation available in the Nigeria which actually used by the Nigerian community for to overcome from the diabetes?
Yes, sir. We have formulations. They may not be standardized. Personally, I also formulate herbs for people to use, and they have really reported the efficacy of the formulations. And we have some other formulations, even in Nigeria. We have several traditional medicine practitioners which formulate herbs for people to use, and people have been saying these herbal formulations are taking care of their disease conditions. So we have them, not in some are standardized, some are not standardized. But the problem we have is hygiene. So that's why we are training the herbal practitioners to be hygienic with their formulations. And we also go to them to seek for the formula. But the problem with them is secrecy. There's secrecy towards this traditional medicine practice. They wouldn't give researchers their information, except to buy their product. And you check the label, and they may not even put all the constituents in the label. That's one of the challenges also. So there are a lot of what is it called? A lot of formulations in Nigeria, actually, as you have in India.
Yeah, yes. One more request is from my organizing team. Although because of the pandemic situation, we shifted the offline mode of the first ICTM to the second ICTM as an online mode. So there is a request from my organizing team as well as from my SMBT Educational Seva Bhavi Trust. The third ICTM is going to happen in the next year once again, and I would like to give the invitation in front of all the audience. You have to attend that third international conference
Okay.
at my institute. So,
Okay.
okay, thank you for
Yeah.
accepting the invitation.
Yeah. Thank you very much.
Put the days towards the our coordinator, just Suryoshi.
Yeah.
Thank you so much, sir. Professor, we have one question from the participants.
Okay.
I will read. So, which type of method used for isolation of active constituents from plant? This is the question.
We have several types of methods to use. I know one of the speaker must have presented to isolate or not to isolate. I saw the program. It depends on the type of isolation you want to do. We have several types of isolation. Are you isolating pure compound? Are you isolating fractions? So we have several types of methods to use. You have to start from extraction from the bulk of the medicinal plant. So from extraction, you have to do partitioning. At times we do bioactivity guided isolation. So when you extract, you check. When you partition, you check. So you check the active partition. The most active one you fractionate. When you fractionate, you refractionate and you check until you get your pure compound. So it depends on you and on availability of materials and equipment to you. But the conventional way is bioactivity guided isolation, so that you not lose track of your activity. When you extract using different solvents, water, ethanol, methanol, or what have you. So you check which of these solvents give the best activity, actually. So you take from there, you check the best activity. Okay, this, for instance, aqueous extract gives the best activity. Okay, let me partition using non-polar solvent and polar solvent. Then you check the partitions again to see which of these partitions give best activity. Then you take this partition, you do column chromatography to check different fractions. And if you are lucky, you may get distinct fractions. So these distinct fractions may be also checked using several assay methods, alpha amylase, alpha glucosidase, or what have you, to check the activity of these fractions. Then you can check if you have pure compound or not. And pure compound is very far, because the complexity of medicinal plants makes isolation of pure compound cumbersome. So you have to you have to waste several kg of plants, and you have to reduce, separate before you get your pure compound. I hope I've been able to do a little justice to that question, please.
I think this was the satisfactory explanation for this question. Is there anyone who want to ask anything, any doubt regarding this presentation? Please go ahead. We have one participant delegate. He is a judge actually for today's presentation. I request Dr. Dolas sir kindly ask one question, please have a doubt.
Good afternoon, sir.
Good afternoon, sir.
As we have a traditional Ayurveda system in India, can you just highlight or share your thoughts on the traditional system which is used in Nigeria?
Okay, sir. Yeah, in Nigeria also we have traditional system of medicine and is as old as our existence too. But there is a kind of mixture into our system. There is mixture of occultism. Those traditional medicine practitioners use not only plants, they use several other materials from animals also to complement the usage of plants. And they use incantations, which is kind of like poem, to make their preparation work effectively. That's the Nigerian system of traditional system. That's why we distinguish between traditional system of medicine and herbal medicine. There are two different things. Herbal medicine use solely herbs.
In addition to the same question, sir, are these traditional medicines official into your pharmacopoeia of Nigeria?
Not totally, not yet totally. We have NAPRID. They are doing a very good work to integrate this into our medical system, and I hope in nearest future it will be achieved. You know, our people, most of them do not appreciate herbal medicine. Is now that the awareness now is getting to the elites. Now, if elites appreciate things will go smoothly because they will be the one to give the approval. So that's the problem, but we are moving very closer to that. Thank you very much.
Thank you, sir.
Thank you so much, sir. Thank you so much, Professor. Yeah, I think questions are got over. I am thankful to Dr. Adiyami who accepted the invitation from the second ICTM 2021 on behalf of entire SMBT Educational Trust and ICTM 2021 organizing committee. I'm thankful to you that taking out some time from your busy schedule and you gave us a nice information regarding traditional formulations. We all the best you sir for your research work in the future, and we are expecting your presence, physical presence for the next year, next ICTM. Thank you.
Okay,
Thank
sir.
you.
Yeah, I'm sending a file, the one I sent to you, Dr. Shir.
Yes, of course.
I've sent the file to the chat for anti-diabetic plants as reported by several researchers. It may be useful for researchers also. Thank you.
Okay. Thank you so much, sir. Have a good day. Do you want to show that file?
Yeah, I've already shared it in the chat section.
Okay.
And if you want my presentation also, I may share now with you.
Okay. Okay. We'll share it with the participants.
Okay.
Okay. Thank you so much, sir. Thank you. Have a good day.
You too.
We have a announcement for the next session, presentation session announcement for all the participants. That session will start sharp at 2 p.m. The updated notification has been sent to your WhatsApp group, respective WhatsApp groups. Kindly connect with the link that we have provided. please adhere to the stick to the timing that we have allotted. Thank you so much. At 2 p.m. we'll meet again, and after which will be followed by valedictory at 4:30, 4:30 p.m. Okay, thank you so much.